Hello, Science 10, and welcome to our very first biology. We are going today to talk about cell theory, abiogenesis, and biogenesis. So today, what are you learning? Well, you're learning about cell theory, which is essentially the development of understanding cells. What makes something living and non-living? We're going to look at animal, plants, bacteria, prions, and viruses. And we're going to talk about who contributed and made it all make sense along the way, from Aristotle all the way to gene mapping and the human genome. So think back, grade 8. Yeah, grade 8 sounds about right. You learn about the characteristics that make something living. Do you remember any of them? Do you have any idea what they are? If you do, hold on to them because we're going to talk about them. But what characteristics do all living things have in common? Wait, what makes something essentially alive? Well, if you were thinking about these 5 things, you are correct. Characteristics of living things. There are 5 things that are really, really, really important. And this is what defines if something is alive. It needs to make or obtain energy. Okay. Some shape or form of energy has to be able to produce wastes. So it has to be able to produce and remove those wastes. So waste can be something like carbon dioxide in our bloodstream heading to our lungs so we can breathe it out. It can be feces and urine. It can be as simple as something small in a cell, a waste product it's made and it's being transported out. But we have to be able to produce and remove waste. Responding and adapting to the environment. You have to be able to understand something that's going on and adapt to that. So you learn about this in Science 9 when we talk about adapting to environments and then learning about evolution and stuff like that. But we respond to things around us and adapt to those. We have to be able to reproduce and produce offspring, and we have to grow. If something is living, it will display all of these characteristics. It's really important to understand. It's not 1 or 2. It's not maybe, oh, I have 4, but not 5. It is all 5 things. So let's look at living versus non-living. Let's talk about the living category first. So in your notes, you have a little chart. So you're just going to add in a little picture perhaps and some information. So in this one, plant cell, I would just draw a square box that looks a little something like this, just to add a few things. We're going to go into a ton of detail about plant cells, so I'm not going to get into it very much right now. But a plant cell, ladies and gentlemen, surprisingly is found in plants. Okay. An animal cell, again, I'm not going to go into a ton of detail here because we're going to go into a lot of detail later, but animal cells are found in humans and animals. Okay. So there's 2 things that are living. Another thing that is living that most people don't think about are bacteria. Now, this is a little bit more because we don't really get into bacteria very much, but I want you to understand some stuff about it. So draw the little bacteria you see there, and let's add in some information. So bacteria, unlike us, are single-celled organisms. Now, they are doing all of the living things that they need to. They produce asexually. Okay, so they can go and they can reproduce by binary fission. They do something called sexual reproduction through conjugation. It's not really true sexual reproduction because there's no offspring being created, but there is genetic recombination occurring, which just means that they become a little different from each other. But if we're looking at reproduction, it's asexual reproduction. Now, some bacteria, not all, can do both photosynthesis and cellular respiration. Now, if you remember, photosynthesis is how plants receive their energy. They make their energy using the sunlight. And we're going to talk about those key structures that are used in that when we talk about plant cells. And then cellular respiration is something that both plants and animals do, and it's how we as humans obtain our energy, or what we call ATP. It's really important. It's a really, really important molecule, and that happens in our animal cells. But cool thing is bacteria can do it too. They grow and then they divide, and that's essentially how they also reproduce, which is kind of fun. Again, they adapt through conjugation. So I already said conjugation is one of the ways we would say sexual reproduction occurs, but the truth is it's really just adaptation because they're sharing genetic material between 2 bacteria to make sure that they can survive, and it changes their genome a little. They respond to stimuli, such as if they think something is dangerous, they can use their long flagella, which is the tail at the end of it, and they can kind of swim away from it because they are detecting some sort of danger. And then the other part is they have a cell membrane that maintains homeostasis, which is really important when we talk about cell membranes. I'm going to talk a little bit more about bacteria. So here's our living category. Now, we're going to contrast this with non-living, okay? So there's living. Let's talk about non-living. You probably have overseen this, and you've probably seen this time and time again, but there's the coronavirus. All right, viruses. Oh, just do this, guys. Sorry. So viruses actually fall into the category of non-living. Most people think that viruses are living, but the fact of the matter is they do not do all 5 things that require us to be living. They can't reproduce, and they cannot create energy. So what happens is when a virus enters into your cells, rather than it being able to divide on its own, it has to enter into your cells and it hijacks your cell's reproductive method, which is essentially asexual reproduction or mitosis. Okay. And so when the cell divides, the virus can divide within the cell, but the virus is not dividing on its own. So it doesn't reproduce on its own. Also, viruses are these little things that float around and they don't consume or use energy. So because they don't consume or use energy, they are also not living. The thing about viruses is although they don't meet all 5 requirements, they do meet some requirements. But because there's not all 5, they fall in the category of non-living. Now, my favorite one, which we're not going to talk about at all in Science 10, but I love to bring up, is something called a prion. Ladies and gentlemen, prions are amazing. Essentially, a prion is non-living, even though it causes a horrible, horrible thing to happen in your brain and essentially results in death. It is not a living organism that is attacking you. It is actually your own protein that folds funny, and then all of a sudden your brain gets filled with holes. That is the simplest way to put it. It doesn't get filled with holes, but essentially the prions will unfold, refold in a way that causes diseases, and those diseases can end up killing you. And prions are fabulous. So I hope you join me sometime in Bio 20 or 30 when we will get a chance to actually discuss prions. So because these guys don't require any energy, they don't reproduce, they just fold and unfold in the case of the prions, they fall under the non-living category. Now that we've kind of covered what is living and what is non-living, let's talk about how the idea of living and non-living came around. So this is abiogenesis versus biogenesis, or the start of cell theory. So a long time ago, 384 BC to 322 BC, Aristotle, one of the main players in everything we talk about, essentially said spontaneous generation occurs. Spontaneous generations means abiogenesis. It means that living things come from non-living matter. Weird, right? What's going to happen is I'm going to play this video for you. Well, I'm not going to play it right now. You're going to watch it and then you're going to move on. It essentially explains what abiogenesis is and spontaneous generation. So now that you have a little bit of a better idea of what abiogenesis is, or how things just magically appear from matter, okay, we have to talk about how did they go from thinking this was something to actually understanding cells. So in 1668, Francesco Redi, yes, very cool guy, performed an experiment because he was not sure that abiogenesis was real. He just thought like, okay, yeah, we say this, but is this actually real? So what he did is he tested whether or not flies could spontaneously appear on rotting meat. So you have to remember flies come from maggots. Flies lay the eggs that we can't see. Maggots come about from those flies' eggs, and then flies are born. Okay. But what they just saw was this random maggots appearing, right? So what he decided to do is he placed 3 pieces of meat in different jars, and he sealed one jar completely. And then he left the others sealed and open. So he covered one completely so nobody could get in. Then the other one was like a gauze covered or a cheesecloth. And then there was one that was completely open. Now, Reddy is amazing because you have to understand that Reddy designed a pretty dang good experiment. He essentially followed the process in which he created variables that he could actually test. So he created something called, well, he didn't create, but he did something called controlled variable. Okay, if you remember back from Science 9 or any other science class, we talk about variables as when we perform experiments, ways to look at our data to see what is occurring. So a controlled variable means that it stays the same or is the one that's not going to change. It's the one that we're trying to keep constant. So what that is supposed to say is his control variable was the meat source and his sterile jar. He used the same type of meat and the same type of jar. That was his control. Okay. If you change the type of jar, then all of a sudden you could have a whole other variable on your hands. So we kept all the jars the same, all the meat the same. His manipulated variable, though, was whether or not the jar was open, slightly open, or sealed off completely. And then what he was looking for, or his responding variable, was the maggots. And what he discovered is when he sealed the jar, surprise, surprise, flies couldn't lay their eggs and then they couldn't make maggots. So he realized that abiogenesis actually might not be something. But unfortunately, it took a really, really long time for that idea of cell theory to take place. Even though he did that in 1668, a couple years earlier, this guy named Robert Hooke in 1665 was actually using his version of a little tiny microscope he kind of created. It's called a compound microscope. Had two little lenses, and what he was doing is he was taking cork, like cork from a bottle, like and stuff like that. It is made out of plant cells. So what he did is he sliced it really thin and was looking at it underneath his microscope, and he was observing what he thought was dead and would put cells because he didn't know what to call them because they didn't have a name for them yet. Essentially, in the end, now that we know what a cell is, he is given credit to actually discovering the plant cell. And his work with the microscope, looking underneath it and trying to look at things that were so small we couldn't see, actually inspired many other people to do the same. One of the main people is the person we essentially say designed the microscope, and that's Antony von Leeuwenhoek. In the 1630s, he started fiddling around with this idea of microscopes, but it wasn't until after he saw Hooke's experiments that he really got interested in microscopes. And when he did, he designed a microscope that he could magnify things up to 500 times. And what he did is he took some water that he found just on the ground and he put it under his microscope. And when he looked at it, what he saw was these things that he called animalcules. And animalcules were just little tiny bacteria. Okay. But again, he didn't know they were called bacteria because they didn't have names. Unfortunately, even though he saw these animalcules by looking at water, and he also like scraped his teeth and looked at what was under what he got from scraping his teeth, although now technically bacteria was discovered, scientists still thought abiogenesis was a thing. They thought these little bacteria were just erupting from nothing. Okay. So even though we now know there is bacteria, or what we now call bacteria, they still believed in abiogenesis. So it wasn't until Pasteur and his renowned um, experiment, which essentially kind of convinced everyone that spontaneous generation no longer could occur. We're going to watch this video. I'm going to post it in the thing and only watch the video until 1 minute and 40 seconds, and then we will move on. So what did Pasteur do? Well, after watching that video, I hope you have a little bit of a better idea, but essentially Pasteur's job was he essentially thought abiogenesis is not a thing. He was like, it can't be a thing. So what he did is he actually was looking at creating an experiment in which he was trying to figure out if microbes would just grow randomly. Okay. So he took what we would call a curved neck flask. He put a broth in it that had nutrients in it. Okay. And what he did, each of his broths that he made, because he had to have a control and one that was not a control, each of the broths that he made was boiled to kill the microorganisms that might be in it. Okay. And then, okay, one of them was tilted so that the neck didn't allow air to get into the actual flask. And the other one, okay, was tilted, allowing stuff to enter the broth. So if you're looking at it here, this is the one that he left straight up so things couldn't get in. And this is the one that he let things get in. Okay. What you have to understand is this one that had the neck laying down, organisms were able to get in and then they grew in the broth. Okay. Oh, sorry. So what he ended up doing was he ended up designing a sterile environment to show that if you kill microorganisms and keep it sterile, that things don't just grow out of nothing. So again, here's another quick video on just kind of more of the wacky history of the cell theory. We're going to watch this quickly, and then we're going to finish off our notes for the day. So now you understand a little bit about who helped make this idea of cell theory by essentially saying abiogenesis isn't true, so spontaneous generation. So we started with Aristotle saying yes, then poof, Reddy happened. And he's like, I don't really know. And then there was Van Leeuwenhoek, and there was Robert Hooke, and then there was Pasteur. Okay. But it wasn't until Matthias Jacob Schleiden and Theodor Schwann that they really had the first principle of cell theory knocked down. And what they said was all organisms are comprised of one or more cells. However, they thought, again, that new cells were created by abiogenesis. So although we have cell theory developing, this idea that there's tiny cells, they still thought, wow, cells just magically appear. It wasn't until Rudolf Virchow that we realize that cells don't just randomly appear, that they actually divide, asexual reproduction, to produce more cells. So after all of this time, we now have a working cell theory. Woohoo! The current cell theory. So cell theory states that all organisms are comprised of one or more cells. We call them unicellular if it's one cell and multicellular if it's more. The cell is the smallest functional unit of life, and I can't wait for you guys to learn about plant and animal cells. They are so amazing, and they are the smallest functioning unit of life. Okay. All cells are produced from other cells. So that idea that cells divide don't just magically appear. So what's kind of happened since then? Those guys were really old. What have we done since then? Well, ladies and gentlemen, a lot. But I'm going to just focus quickly on a few of the things that I think are very interesting. There's so much more out there. But let's talk about in 1953, the discovery of DNA. What? So cells. They knew there were cells. They started to look at cells. Then they started to realize, hey, there's things inside these cells. And what are these things? And then they found the nucleus. And the nucleus contains your DNA, everything that makes up you. But up until 1955, they knew something was in there. They knew DNA kind of had to exist, but they never saw it or knew what it looked like. So they're credited with the discovery of DNA, or the double helix structure that we call this. Now, Watson and Crick were credited with it, but Rosalind Franklin, who was a crystallographer or someone who used X-ray, is actually the reason that they were able to build the model they built, because she took the very first picture of what we would call our DNA, or our double helix. So the world of DNA started here, 1953. That's not that long ago, you guys. Think about it. Yes, it seems like a long time ago, but it actually wasn't. If you think that we started this story with Aristotle way back in like 386 BC or AC, I can't remember what I said. So now we've moved on to so many cool things, the human genome and CRISPR. Okay. The Human Genome Project was started in 1990 and completed in 2003. Okay. And when you watch this video, the Human Genome Project, you will get to learn about how they mapped all the genes in the human body. And ladies and gentlemen, it was not a short feat. It took 13 years, and it is amazing. It is the reason that we can do 23andMe, Ancestry, DNA, and all those cool things. It started here. And then in 2017, something new came out on the board, and that is CRISPR. And CRISPR now lets you be able to edit your DNA. So CRISPR can actually go into the nucleus, go to the DNA, and can find little tiny pieces of DNA, and it can cut and replace it. So things that are simple base pair mutations, which just essentially means these guys right here, these guys are base pairs, they match up. If they match up wrong and create a mutation, you can end up with a disease. If it is a simple mutation, CRISPR can go in, edit, and try and fix it. Isn't that amazing? So this is the stuff that we now have, all because people thought, hey, things just come out of nowhere. But now we know cells exist. There are things inside of cells, and DNA is one of the coolest things that you can find inside your cell. So ladies and gentlemen, I hope that you were able to pull away some things. You need to know cell theory, those 3 points I talked about, okay? The cell theory, 3 points. You need to know that. You need to have a little bit of an idea of who those gentlemen were that came along and made some big, big things, and what makes things living and non-living. Okay? Those are the big ideas. So thank you, and I hope you have a great night.